What did one volcano say to the other? What did one volcano say to the other? I lava you. Nice. What kind of dance do mothers like best? What kind of dance do mothers like best? The mamba. Who said it? Nicely done. If you didn't get it, come see me after class. What goes up and never comes down? Age. Age. Well done. I feel like you all are pretty much ready for the exam next week. What do you think? There won't be any questions like those, but just more of a gut feel. Okay, so announcement number 1. Next week is exam 1 in this room at this time. So here's what it's going to look like. You get a test packet, and you're going to have a slightly different version, actually a very different version than the person sitting next to you. You're going to get a Scantron, and the good news is they're for free. In this day and age, we charge for everything, but Scantrons are included. Okay? If you need a second one, I'll bring extras. Okay? If you need like 5, well, then we're going to have to talk because you need to practice some Scantron skills. Bring a number 2 pencil. It could be a mechanical pencil, a lead pencil. Black ink actually does work, but you can't erase your answer. Okay? I'll probably have a small box of pencils, but they're the kind that, you really can't write with. They're like this big. So if you're in a pinch, I'll have some golf pencils, but try to remember to bring like a mechanical pencil or 2. You're going to have the full hour and 15. I'll let you know where you are as far as time allotment. We'll have Proctor circulating the room. The 4 of us will be the only ones that are allowed to answer your questions because we're in every lecture. Okay? You can ask questions during the exam. Doesn't hurt. The worst thing that we can say is, I'm sorry, I can't answer that exact question. It kind of gives away the answer to the test question, but I can tell you the following. So just try it. 50 questions, closed book, closed notes. You're going to turn off your phones, take out your AirPods, turn your baseball caps around. It's kind of a traditional, like we used to do in the old days exam. Okay? No technology to worry about, just you and the exam packet for an hour and 15. Make sense? There's a study guide. Who's found it already? Study guide in resources for exam 1. Couple things about the study guide. It is not intended to replace studying or coming to lecture. It's there to help reinforce. So it's not a replacement, it's a reinforcement. The way that I would use it is I would probably look at the study guide and find out like, hey, is there something on there? I have no idea what he means. Did you kind of breeze by a topic that you don't have any information on? The best tool is going to be rewatching the lectures or looking at your study notes from lecture, because I take all my test questions from where? From my lectures. All right, we're going to have a participation assignment today. Here's how that works. You get 10 points per unit. So 5 are going to be handed out today at the end of class. It's easy participation. I don't take attendance, but this is how you get participation points. You get 5 today for being here, all the way to the end. You're going to have another 5 points available between now and next Monday by going to one of many study sessions. You only have to go to one, and you get 5 more participation points. These are not bonus points. These are like, there's 10 out of 10 available for Unit 1. If you look at your syllabus, there's 50 participation points, 10 per unit. Okay? So you don't want 0 out of 50. But if you're coming to class and you're studying for the exam, you go to a review session, you should be getting 50 out of 50 participation points. There's no reason that you shouldn't. If you miss one, 45 out of 50, that's still not bad. That's still a 90%. Okay? You can go to any one of the review sessions, and they're going to give out the code for the participation. And it's just a yes or no question, to be honest. It's really easy. Is there any questions about that, the exam format, where to go, what to bring, how to dress? How do you find out when? That is a perfectly placed question. When are the review times?
So I'm going to have an SI session on Thursday from 6 to 7 in Science Annex room 122. That'll be dedicated to exam review. I'll have an office hour on Wednesday, bio room 145.
I'll be holding mine right after class on Wednesday from 5:30 to 6:30. I'll be in bio room.
And then anything this weekend?
Yeah, then we'll have our joint session, all three of us, on Sunday from 3 to 4 p.m. in bio room 256.
Did you catch all that? And that'll be in the announcements. We'll post them in the announcements. Question? The one in Bio 256, that one's going to be streamed on Zoom as well, correct? Okay. So the Sunday option is by Zoom as well. So you have in person and you have one last final Zoom effort on Sunday afternoon. Question? When will grades be posted? 15 minutes after you're done. No, I'm just kidding. Probably by the next evening. Or Wednesday at the latest. There's 180 of you, but we go pretty quick. There is a opportunity for bonus points before exam 1. But I'll talk more about that on Wednesday, and I'll explain how that works. So you have to come Wednesday. Any other questions? Quiz number 4, the last quiz for this unit, is due this Friday by 11:59 p.m. And now we're going to launch into really our first new material for the semester, non-review. We're in histology and tissues. And what's nice about this week is it completely lines up with lab. How many of you, nobody's really had lab yet, huh? So that's nice because this week, today and Wednesday, which is all game on, this week is new material that will be testable material for next Monday. This week in lab, you're going through anatomical planes, microscope, and tissues, and doing histology. And so that's what we're actually doing in lecture this week. So we're like perfectly in sync. And then we'll kind of like move away from each other for a little bit, and maybe come to fall behind a week or two. But right now we're actually perfectly aligned with lab, which is kind of nice. So you'll have it reinforced this week. It happens to be the new material that the first new material for this semester, and also your first full week of lab with real stuff. Okay, so upper left, can anybody tell me what that image is? That's a histopathology image of what? It is connective tissue. I thought someone was just going to say tissue, but you would have been right as well. Connective tissue were even more correct. That's areolar connective tissue. And in lab and in lecture, you should be familiar with what that looks like. Okay? So we'll go over these. We'll talk about connective tissue, but its purpose is to connect stuff together. That's why it's named connective tissue. What about this lower right image? What is that? It is bone tissue. Bone is another type of connective tissue. So different organizations, does it surprise you that they have different architectures? Because they have different what? Purposes or functions. All right, you're starting to catch on. So the study of histology is really the study of microanatomy. The study of microanatomy. So we have to us the ability to look through a microscope now, thanks to Robert Hooke, and many of those that came after him and refined the microscope. So we can look at microarchitecture and get clues about how things work. And that's really all histology is. Histology, microanatomy, histopathology, all of these are similar terms that are used synonymously. And the tissue that we look at is a collection of cells. Remember when we looked at the hierarchy of living things? Cells is where life begins. And a tissue is a collection of cells that all perform the same function. So in our bone example, these are bone cells that all come together to perform a function of a long bone or a flat bone. Long bone would be arms and legs. Flat bone would be like your skull. And then an organ is a collection of two or more tissues that work together to perform a task. An organ. So like, for example, heart has at least two different types of tissue. There's actually a handful or half a dozen different tissues in the heart, and that's an organ. Same with the lungs. Same with skin. Does that surprise you that skin's an organ? And we're going to cover skin or the integument the first week or the first lecture after the exam. So we'll have exam 1 on Monday, and then we'll get into the integument or skin on Wednesday of next week. And what is this a picture of down here on the lower right? Who said it? Neuron, thank you. That's a neuron. The big purple smear is a neuron with the nucleus in the center. And all of these small little cells on the outskirts are called glial cells or supporting cells. And we'll talk more about that later in the semester. Upper right, what kind of tissue is that? It is epithelial tissue. I feel like you might have looked ahead a little bit. I like that. This is epithelial tissue. Anything more specific about the type of epithelium? This is pseudostratified ciliated epithelium, and we'll cover that. And you'll be able to discriminate between different tissue types throughout lab and lecture this week. So let's look at the 4 different types of tissue. We've got epithelia or epithelium. We have nervous. We have muscle, which is the only one that I didn't show yet. And then we have connective tissue at the bottom. So if you look at the embryonic source of the adult examples of epithelia and nervous tissue, so the epithelium and the nervous tissue arise out of the epithelia in the embryo. The epithelium lines cavities and covers surfaces. Nervous tissue, like example, brain and spinal cord. Muscle provides movement, like skeletal muscle or even smooth muscle or cardiac muscle. And then connective tissue brings things together, like our bone example or tendons and ligaments that we'll look at later today. So something to keep in mind with histology is, and this will be helpful in lab, but you kind of need to think about the orientation of the sample that's been collected. How many of you actually ever had a biopsy performed of your tissue or know somebody that's had a biopsy? Usually it's like a suspicious mole or something on the skin or a tag or something that has changed color, shape, size, or it starts to maybe have a little bit of irritation or pain when you move or touch. So we grab a biopsy sample of the tissue, and we take that sample and we process it first. And I mean, if you're ever interested in seeing this, we do this here on campus. We have a facility that does this, a core histology facility that does this on campus. I actually am the director of that facility, so if you want to see it, I'm happy to show it to you. But what we do is we take this tissue and you fix it first. You cross-link it. You cross-link the protein so it stays in place and it doesn't degrade. So use a fixative to do that first. Then you take all the water out and you go through a series of dehydrations, like 70% ethanol all the way up to 100% ethanol. As you increase the percent of ethanol, the water is starting to come out of the sample. Ultimately, you actually embed it in a piece of wax, paraffin wax. And now that tissue can pretty much be preserved in perpetuity. It'll really never spoil, never go bad. And you infiltrate all of the tissue with wax, all the spaces, all the crevices, and then you have a block of wax that has a piece of tissue in it. And then you slice it very thinly, and you can slice it like if you see this sort of noodle structure, like this elbow noodle at the top, and you see kind of the dotted orange line across the top. If you cut in cross section this way, this orange line, you're going to see this orange surface when you look down from the top of two circles. Can you appreciate that? If you go a little deeper into the tissue and you slice here at the green dotted line, and then you look down from the top, you're going to see sort of these two circles, but they're kind of connected because you're right here at the elbow. If you go a little deeper in the tissue and you slice at this blue dotted line, you're going to see what looks kind of like an oval egg. So depending upon where you slice in that piece of tissue, you're going to get different pictures of what it looks like. Similarly, if you look at this squiggly tube, which could be a blood vessel or an intestine, if you slice all the way down coronally, you're going to actually see parts of this and then a gap and parts of this, because this thing is coming in and out of the plane in the Z direction. So in lab, when you start looking at tissue, kind of pay attention to the fact you're looking at a two-dimensional representation of a three-dimensional structure, and you're somewhere in that plane. And this is a similar type of thing that happens with a lot of radiology or imaging. Anybody ever had a CAT scan or an MRI? Same kind of thing that happens. And so as you move through the body getting pictures, you're getting slices in different planes, and then you can reconstruct it with software. But having the ability to see a 2D picture and understand how it reconstructs three-dimensionally takes a little bit of training. Okay? So the stains can be, or the orientation can be deceiving, as well as the staining can be deceiving. So what is this histology picture of? Any ideas what we've stained here? What kind of tissue is that in the lower left? Any guesses? I'll give you a clue. This is hyaline cartilage. By the end of the week, you should get better at recognizing all of these. This is hyaline cartilage. How about this one right here? You can see this. These are actually labeled as chondrocytes. So if they're chondrocytes, what kind of tissue is it? It's cartilage. What kind of cartilage is it? It's also hyaline cartilage. Oh, wow, that's weird. What about this one over here, just to the right? What kind of tissue is that? Hyaline cartilage. Oh, wow, someone's catching on. How about this far right one? What do you think about that one? Looks completely different. I mean, it's black and white. These are pink, and this one's blue. What is that? It's also hyaline cartilage. Okay, so the stains can be deceiving. So the best histopathologists are individuals that focus on the architectural shapes and less about the color. In fact, in the old days, when I was in grad school, I don't know, in the last millennium, peer-reviewed publications with histology images were actually in black and white because it was expensive to publish color. Like if you had a really big grant, you came from like a wealthy laboratory, it's kind of like coming from a wealthy family, like, you know, your PI had lots of grant money. You could afford to get like one color picture, and you chose very carefully which color picture it was. In histology, you would just publish black and white because most people are pretty well trained at just looking at the shapes and the features. So I don't, in lab, that's my tip to you, is try to focus on the shapes as you draw them in lab of what you're looking at under the microscope. Pay a little less attention to the coloration, okay? All right. Also, because I'm going to ask this question to the audience, what color is our tissue? That biopsy sample that we just took. What color do you think, if I sliced a piece of that paraffin wax and I got a piece of tissue that's embedded in wax and I put it on a glass microscope slide and I don't stain it, I just look under the microscope slide and I shine light through it, what color is it? Pinkish? Any other guesses? Transparent? Well, that's interesting. Colorless? Okay. Any other guesses? Does it depend upon our ethnic background at all? You think? If it's skin? Maybe. Now you're like, oh, I don't know. Now that's a trick question. So transparent and colorless is absolutely correct, regardless of your ethnic background. So this is what skin looks like, pretty much unstained. I stained it with a slight methylene blue so you could appreciate the nuclei. But this is skin. This is the stratum corneum at the top. And this is the dermis down here, where your tattoo ink would go. And this is the stratum basale layer right here, the bottom layer of the epidermis that interfaces with the dermis and the epidermis. And the coloration of this individual skin is found by the brown stain that's melanin that are made by melanocytes. But other than that, regardless of your ethnic background, our skin all looks pretty much the same, translucent, clear, colorless. Those of us that have darker skin, maybe a little bit more melanin. Those that have lighter skin, less melanin. But that's it. Kind of those one cell type that differences the color of our skin. Otherwise, everything else is exactly the same. So you actually have to stain the sample to see features. And we stain it with substances known as like hematoxylin and eosin. That's the two main stains that we use in histology. One second. Eosin is going to pick up the collagen and stain it pink. And the hematoxylin is going to bind to dense nuclear material or dense DNA material inside the cell. And so the purple dots are all the nuclei, and all the pink is basically the connective tissue. Question? How? It was collected under informed patient consent with an IRB. Yeah. Great question. Yeah. Not another Henrietta Lacks story, don't worry. Okay, let's move to epithelial tissue. Let's move to histology. So, great question, but like in a lot of clinical trials that you do, and you guys have seen these, like ads for clinical trials, you can volunteer to be part of a clinical trial. You have to actually sign an informed patient consent saying I consent to anything that's being done. In the protocol, you get to read the protocol. Not anything in the world, right? And those clinical trials are actually under the umbrella of what's called an institutional review board. So the institution actually has to have a panel that says, yeah, this protocol can be executed on. You can recruit patients and enroll them in this trial. So we did a lot of work with skin and dermatologists and plastic surgeons. And so the medical side of our colleagues would actually recruit these patients and take the biopsies, and then we would get them, and we would actually process the tissue. Okay, so epithelial tissue. Different types of epithelium. But all of these different types have main features or main functions that are common. Like, for example, protection or barrier, found in the skin and the respiratory tract. Absorption. You're going to find epithelium in the small intestine to absorb nutrition out of the food that you eat as it passes through the GI tract. Secretion, like in the kidneys and glands, those epithelial tissue are responsible for releasing things. And then last but not least, filtration. They'll filter. They serve a filter function like you find in the kidneys. So different types of epithelium have different purposes. Now, want to bet the shape of the epithelium matches up to the type of purpose or function that it serves. Make sense? So if it's more like a cube or a square, it has either more of a secretion or absorption or a filtration. If it's flat, we have a term for that. We call it squamous, which means like flat-like or plate-like. Then it forms like a sheet or a protective coating like armor. And then you can stack these flat plates of epithelium on top of each other, like what we saw here. You've got a lot of epithelium that's stacked on top of each other. That's the outer layer of your skin. And that squamous, simple squamous epithelium, as you stack it, it becomes stratified. Simple is one layer, stratified is multiple layers. So that outermost layer of the skin is a stratified squamous epithelium. Nice and protective. Cuboidal, simple. You stack it, it's stratified. So the stratified terminology goes to multiple layers. The simple terminology goes to a single layer. Simple columnar, and then you don't have a stratified. You have what's called pseudostratified, which I showed you. Pseudostratified columnar epithelium is always ciliated. It has these little cilia on top. Why do we call it pseudostratified? What does pseudo mean? What's that prefix? Not real, fake, false. The best translation would be false, but all those would be synonyms, okay? So it's falsely stratified because if you look at the basement membrane at the bottom, all of these cells, this one's kind of wide at the top and then narrows and touches the basement membrane. This one's wide at the bottom, then narrows and peters out. So all of these are attached to the basement membrane. In true stratified, you actually have layers that are stacked on top of each other, and the second layer doesn't touch the basement membrane. That's how you know that it's stratified versus falsely stratified or pseudostratified. The last one's called transitional. Transitional has the ability to go from like a cube and flatten out, like when you expand and stretch. You only find transitional in one organ, and that's in the bladder. And I'll show you some pictures of that here in a little bit. Okay, so epithelial tissue. The characteristics. So here's a thing that's probably good to memorize. Epithelial tissue is cell-rich and matrix-poor. It has a lot of cells, and it has less matrix, less outside of the cell, less connective tissue, less collagen. It's very, very densely packed with cells. So we say cell-rich and matrix-poor. And these cells are tightly packed together. They're tightly packed together with these, what we call tight junctions or adhering junctions. And they communicate next to each other. Like their plasma membranes have these proteins that actually talk next door to each other. These adhering junctions have another term known as desmosomes. That's the fancy literature word for these adherent junctions or tight junctions. And oftentimes, epithelial tissue is relatively avascular, or there's not a lot of space for blood vessels. So they don't have a very rich blood supply. But the diffusion distance of oxygen in tissues, like just the simple diffusion from a high concentration to a low concentration, is about 250 microns in distance. 250 microns. So epithelial tissue typically will have a blood supply that's less than 250 microns away in some other adjacent tissue, because the blood vessels don't come into the epithelium. They actually are deep. So for example, you guys know that you can scratch your epithelium on your skin and it may not bleed, right? You can actually, I don't know if you ever did this, like when your kids or whatever, did you ever put like straight pins like through the calluses in your hands and then like try to scare the person next to you, right? I know. It's weird, I know. But some of you know what I'm talking about. So you put a straight pin through like the callus and or thick parts of your skin, and it doesn't bleed, right? There's no blood vessels there. So the vessels are usually found in the underlying connective tissue in the dermis. And if you get a deep enough cut that goes below the epithelium, then it starts to bleed. So cell-rich, matrix-poor. Now, they also are polarized. That means they have a side. Polar means it has a side. Like the phospholipid bilayer, which is polarized, right? It has a side to it. So the basal side, or the basement, is supported by the connective tissue. That's where the cells attach, on the basal side. The apical side is usually exposed to a lumen like what you see here. So here's a squamous cell. Do you see how it's nice and flat? Here's a simple squamous layer of cells because they're not stacked on top of each other. And the basement side is the side that's touching the green stain. And the apical side is sitting in this lumen. And those are actually endothelial cells inside of a blood vessel, simple squamous epithelium that lines the inside of a blood vessel. So the apical side is the lumen side that faces out. It's the site of secretion. It may have the microvilli or the cilia, like what we have here in a pseudostratified ciliated columnar epithelium. And the apical side here is going to be the lumen that you're releasing substances onto. And this lumen you're releasing substances onto, you release a mucus. This is in the respiratory tract. You release a mucus out here via these goblet cells. And the cilia and the mucus help to catch any debris as you breathe it into your respiratory tract, catches the debris, and then brings it out so it doesn't sit in your respiratory tract. So every time you clear your throat, those respiratory epithelium move through this respiratory escalator, move debris and mucus up and out, right? And helps you form what you conveniently refer to as a loogie, right? That's how that works. So exocrine, releasing onto lumen or outside. Exo meaning like exiting the body. Endocrine, inside the body. This is releasing it into the bloodstream. It's usually ductless. And so you might see releasing into the bloodstream example with a simple squamous epithelium where substances are released into the blood supply. Additional characteristics of epithelium. Well, they have a rapid ability to regenerate. The cell divisions are quick. At the basement layer, the basal side, is where they typically have their regenerative layer. And they'll replicate and make another cell, and that moves to the apical side or to the superficial side. So take the example of skin. The basal layer or the basement layer of the epithelium is where skin regenerates from. And then that new cell moves superficially towards the surface. And as it gets further and further away from the blood supply that's in the dermis, it becomes less and less oxygenated. And so the cell starts to slow down its metabolic activity. As it does that, it actually starts to pack up its organelles and gets rid of its nucleus. And the cells die as they move superficially away from the blood supply. And then ultimately at the top, they slough off on that stratum corneum layer. And that turnover that takes place happens about every 6 to 8 weeks in you and I. We're slowly sloughing off the superficial layer of our epidermis, known as the stratum corneum. That's a vast majority of what dust in your house is made of, right? And you regenerate it every 6 to 8 weeks from the basal layer of the tissue. So if you look to see this bottom of this patient's heel, you can kind of appreciate this layer that I'm talking about. Here's the connective tissue that's deep. Here is this basement membrane. This is the regenerative layer, and we move this direction. These are living cells. As it moves further away, it gets to this nice delineation, which is almost exactly 250 microns away from a blood supply. And now these cells are dead. They're dead and they're squamous. You see how these are more rounded and these are flat. And in this flat zone, this is where you're sticking that straight pin. You stick it too deep, right? Your friend goes, ah, ha, ha, that's funny, and stabs you. Then it bleeds, okay? As long as you're surgical, as you horse around as a kid, you can target this squamous cell, and that's how you know that you're in the dead tissue. The need to regenerate is because our skin's under constant stress. And most epithelium is, but the skin especially is under constant stress in our environment. Okay, different types of epithelial tissue. If we look at the two that I wanted to highlight, pseudostratified and transitional. This pseudostratified, here's another picture of pseudostratified. So you can kind of appreciate that here's the basement membrane. Here's the connective tissue that's down low. Do you see how these purple nuclei are further apart and these purple nuclei are stacked right next to each other? So the epithelium is cell-rich, matrix-poor. Guess what? The connective tissue that we're going to get to is just the opposite. It's matrix-rich and cell-poor. So you can really nicely appreciate this delineation between epithelium and connective tissue below it if you just look at the shapes and the architecture. So the pseudostratified, it's always ciliated. A lot of times there's mucus that's being secreted like we talked about, and the cilia sweeps it away. And then transitional. So this is the bladder. So these shapes, here's the nuclei in the center, that round darker stain. And then around it is kind of this halo of the cell itself. It kind of looks more like a fried egg, in my opinion. Okay, but it's actually more cuboidal. Okay, it's kind of a cuboid-ish shape. But think of like a fried egg as you're looking down at it. And this is in an empty bladder. And then here, as the bladder expands, those cells actually flatten out into more of a squamous architecture. Now, they're not going to get fully squamous until you're like, your eyeballs are floating and you got to run to the restroom. But any time during the day, as your bladder is filling up, because your kidneys are constantly filtering your blood and making urine, and the bladder stores it until there's a convenient time for you to actually empty it. So this is the transitional epithelium when it's full, and you can see that these cells, it's easier to see them towards the basal layer, but these are definitely flatter and not as rounded, and that's what you would see with a transitional example. Only found in the bladder. So this is off of that histology DVD that's like part of your lab material. It's optional, but this is where we got this picture from. And so this is the bladder. This is the area that you would see the transitional epithelium. And beneath that, this is all connective tissue. So here's our connective tissue. Here's our transitional epithelium, connective tissue, so on and so forth. So this whole space in here that's the area of the bladder that's going to fill, and it's going to make these structures kind of flatten out like a big balloon, right? Imagine a big balloon inflated. All of this white space turns into a giant circle. So functions of simple epithelium, and then we're going to talk about the functions of the other types of epithelium. Simple, absorption, secretion, diffusion. Simple, absorption, secretion, diffusion. So simple cuboidal or columnar lines the digestive tract and the kidneys. The pseudostratified, we said this a few times, this is always ciliated, and the cilia helps sweep the debris away. So on the lower left, we've seen this picture before. What type of epithelium's on the lower left image? Simple squamous. Very good. This happens to be out of does anybody remember where this came from? Does anybody remember where it came from, this sample tissue? That's of a blood vessel. The middle one, what type of epithelium? Simple cuboidal. Simple cuboidal. So if you follow these rings, you'll see they're made up, like the darker circle is the nuclei. They're made up of cuboidal cells that are arranged, kind of like a pineapple slice is what I think it looks like. And they might be slightly wedged because like a pineapple slice, on the outer side, it's a larger diameter, and the inner circle is a smaller diameter. So they're not going to be a super perfect cube. They're going to be kind of, right, more of like that. The simple cuboidal picture you find in the kidneys and the kidney tubules. And then the lower right, what type of picture is that? Pseudostratified ciliated columnar epithelium. It's a mouthful, but every one of those terms helps to describe what it does, right? Pseudostratified ciliated columnar epithelium. And that one's found in where? Respiratory tract. Perfect. Respiratory tract. Okay, stratified. Stratified usually is for protection, like what we talked about with the skin. So you'll find it the skin, the inside of the mouth, the cheek, the esophagus as food comes down, the anus, the vagina, all of these places where there's potential for abrasion, that's where you're going to have stratified epithelium. And in all of those cases, it's essentially stratified squamous epithelium. The basal side, like we talked about with the skin, is the regenerative side. And as it moves superficially, it sloughs off, and then it regenerates and replenishes. The apical side is the side that dies and sloughs off. So you can see a couple of different pictures. Lower left, this is another example of pseudostratified. So it's really not the stratified for protection that's found in the respiratory tract. But I want to make sure that we give attention to pseudostratified because it's really not simple and it's really not stratified. It's sort of this in between, right? I guess technically it's more simple than anything, but we call it falsely stratified. So just showing you this one next to truly stratified epithelium. And in the middle picture, another pseudostratified. And then this is skin. This is the stratified squamous, where you've got the basal layer here. You've got the apical layer here. You can see the nuclei, the dark stained purple nuclei, circles or ovals, get fewer and fewer as you move superficially as they slough off. So true stratified squamous on the far right, two examples of pseudostratified in the middle and the far left. Questions on epithelium? All right, let's transition to connective tissue. Today we're going to talk about epithelium and connective tissue. Then on Wednesday we'll come back and we'll finish out histology and tissues with the rest of the material. So connective tissue, what is its purpose? What's the purpose of connective tissue? Give me some ideas. Connects things to other things. That's beautifully stated. Anybody want to say it a slightly different way? Because there's nothing wrong with that. That's perfect. Keeps our bodies from falling apart. It's like the glue. It holds stuff together. So it's really important. Now, the epithelium is important too, right? We just got done talking about all these different things that epithelium does. But you have to have it bound to the rest of your body, and the glue that holds it together, keeps it from falling apart, that connects it, is connective tissue. So it's determined by the cell and the matrix type. Connective tissue can be a little bit more challenging for students because there's more categories and variations of connective tissue. I think epithelium is a little bit more straightforward. So do epithelium first, you nail that, you're ready to move on to connective tissue as you study. You might find it a little bit more challenging, but the repetition, you'll get there, I promise. So it glues things together, it binds things together. If it's bones to bones, it's a ligament, and ligaments are a type of connective tissue. If it's muscles to bones, it's a tendon. You know, because your muscle is going to contract and pull on something, but it has to be attached to be able to pull on it and generate force. That's a connective tissue that attaches a muscle to a bone. That's referred to as a tendon. It glues these different types of tissues and organs together so the organ doesn't fall apart. Now, there's a couple of different types of disorders that are connective tissue disorders. Has anybody heard of Marfan syndrome? Okay, a couple of heads have nodded. So Marfan syndrome is an abnormality in the development of connective tissue, key connective tissue. Tends to be collagen and elastin, depending upon the Marfan syndrome. You can Google this later. This isn't on the exam. This is just information for your mind. So these patients, unfortunately, don't make connective tissue as strong. Okay, if that's found in the blood vessels, like the aorta, that are highly pressurized, there's a lot of force in the aorta over time. So then the aorta starts to enlarge and dilate, and it can rupture, and that's often how Marfan's patients pass, is through an aortic aneurysm or a rupturing of the aorta. Okay, so just an example, a clinical example of why this is important. Okay, now that is not a picture of an aorta, Keller. It's somebody's back. What's happening there? Okay, so this is a process known as citricization. And it's inducing remodeling of the connective tissue in a very specific way to induce scarring in elaborate patterns. This is a French term. Citrice is what the French origin term comes from. But it was describing some native tribes in Africa that would do this as an identification of who they belonged to. Okay, a type of, you guys have seen Black Panther, right? Where Michael Jordan had intentional scarring that he had on his torso, right? Looked pretty awesome, right? But that's the remodeling that takes place in connective tissue. So when we come back after exam 1, when we start talking about skin, and we talk about wound healing and skin, we talk about scarring. So it's not just the epithelium that has to heal, which actually heals really well. It's the underlying connective tissue that has to heal too. Okay? And as it heals, it may not heal in its original embryonic development form. And so as it heals abnormally, you get scarring like this. Okay? But that is a function of the connective tissue being able to do what it's supposed to do for all of us. So some of the functions of connective tissue, and just categorically, we use it in our bodies to support and protect. We use it to cushion and insulate. So we have connective tissue around organs, and they form a capsule because those organs are important. It's like, like an Amazon box, and you open it up and there's like little white peanut packings in there, or there's like, paper, that brown paper, or, if you get one of these newer Amazon boxes that use 99% less everything, and you open it and your thing's broken, right? You're like, I wish there was more like cushioning support there. So not something that we want to skimp on in our bodies, right? Supporting and protecting. Bones help to support and protect. You're going to, in lab, you're going to start getting into like the appendicular and the axial skeleton. You'll start studying the rib cage. The ribs protect our vital organs right here, right? They end about right here, and on the back, they end about right here. But for the most part, the heart, the lungs, the vital organs are actually protected by a rib cage. Bones are a type of connective tissue. All right, cushioning and insulating. Within an organ, there's loose connective tissue that helps to cushion. We find this like in the skin. And then adipocytes, that's just a fancy word for fat cells. Fat cells, adipocytes, insulate and cushion around all of our organs. If you've ever been in a OR, or you have an opportunity to see like an open chest procedure, you're going to be shocked. What color do you think the heart looks like of a 75-year-old going through a bypass procedure? When you open up the chest, what color is the heart? Gray? It's not gray. It's a good guess because you're thinking, oh, it's probably disease, so it's not going to be. Who's going to say red, right? Valentine's Day is a few weeks away, right? Red hearts, come on. It's yellow. Because there's so much fat that sits around the heart to protect it. Fat in our bodies is a yellow color. It's not white. Most people think it's white. It's actually yellow. So adipocytes or fat cells are a type of connective tissue. Other things, stores and transports nutrients. Like adipocytes store fatty acids. Remember, in our prior lecture, we talked about energy sources, and fatty acids, amino acids, and sugars are all three energy sources that our body uses. So your fat supply that you store, when you need it, you can recruit those fatty acids and run them through energy pathways to generate ATP. That's why we store fat. For those in between, you can't get, to the vending machine quite fast enough, right? You need something, you have it with you. Adipocytes, bone marrow, blood cell precursors. They store where our red blood cells come from. They store where our white blood cells come from. We kind of alluded to that in one of our earlier lectures. Bone stores calcium. Ca2 plus is calcium from the periodic table. We'll talk about the role of calcium, which is essential in muscle contraction. When we get to our muscle physiology unit and we start studying muscle contraction, we're going to focus on calcium and calcium's availability. You cannot get muscle contraction without calcium. Did you know that? That's why it's so important. But it also is important because the bones store it. The bones can release it when it's needed, but they store it, and they store it in a calcium mineral that forms a hard substance, and that's why our bones are the kind of density that they are. Blood. So fat, bone marrow, bone, blood, these are all types of connective tissue. Blood transports nutrients. Blood is a connective tissue. Doesn't that blow your mind? Because it's liquid. You're like, it's liquid, Keller. Yeah, until you have a cut and then it forms an elaborate matrix as a clot to prevent the bleeding. Okay? It's a phase-changing connective tissue, which is kind of amazing. Sounds like a sci-fi movie, in my opinion. And that circulates in your body right now, your connective tissue. And patients that have clotting diseases or clotting deficiencies or issues with clotting very well understand the importance of this connective tissue being able to phase change appropriately. Lower left, this is a picture of what? Bone. These are all connective tissues. So this is bone here. Middle, bottom middle, red blood cells. Bottom right. Oh, it's tougher. You probably haven't seen it yet. That's bone marrow. That's bone marrow. So the arrow is pointing to components or spiculars of bone known as trabeculae. And then all of the open space that's kind of more of a white open space. This is actually primarily adipose tissue, fat tissue in the bone marrow. And this is where a lot of the red blood cells are generated within that adipose tissue. We'll talk about that more this semester. It's called hematopoietic bone marrow. Upper right, what's that? Adipocytes, fat cells. So they're really kind of fascinating because they're like storage silos of fatty acids or energy. And so most of what they are is this open white space. You see the single nucleus is like smashed up against the edge, right? Because it really wants to use all of that open vacuole space to store fatty acids and no water. That's the benefit of fat. It's a very efficient storage form of energy because it doesn't like water. It's hydrophobic. So water is extremely heavy. And so if you have to carry a lot of energy around in a lot of water, water is precious. You need it other places. And it's actually heavy to carry around. So your muscles and your bones would have to be bigger and stronger. So you use fat, you get rid of the water, you store the fatty acids, and you can still get through aerobic respiration to make ATP. It's actually really clever. So imagine these are just like, imagine like the Midwest when you drive through and you see like these giant silos of, grain silos. That's what fat looks like to me. Like from a downward view, just like silo after silo right next to each other. They look like kind of like balloons all together when you look in lab. Okay, characteristics of connective tissue. Remember I said cell poor, matrix rich? So this picture is what kind of connective tissue? We've seen it before. The very beginning. Upper left picture, areolar connective tissue, right? You can see I've labeled a couple of fibers here. You can see the pink, larger diameter fiber is a collagen fiber, and this black dark line is an elastin fiber, thin, right? This one's a lot broader. The cell pore, the immature cell in connective tissue is usually referred to as a blast, like an osteoblast or a chondroblast. Osteoblast is an immature bone cell. A chondroblast is an immature cartilage cell. If it's mature, it's referred to as a cyte. So like an osteocyte is a mature bone cell. A chondrocyte is a mature cartilage cell, cartilage producing cell. And then they're matrix rich. So there's more matrix. You can see in this picture of areolar connective tissue, now hopefully you can kind of appreciate, okay, so that's a nucleus, that's a nucleus. These dark, spheroid looking structures are nuclei, and there's not that many of them compared to epithelium. There's a lot more other stuff. There's a lot more connective tissue. There's a lot more matrix than cells. So components that are outside the cell are referred to as the extracellular matrix. So in connective tissue, the matrix or the ECM is abundant. And that's mostly what holds us together and keeps us from falling apart, is this extracellular matrix. And the primary dominant protein in the extracellular matrix is collagen. So when we're talking about cell biology, I use collagen as an example that you manufactured inside the cell, and then do you keep it inside the cell, or what do you do with it? You excrete it. You exocytose the vesicle that holds it, and it is released out into the extracellular matrix, and then it actually starts to form. So characteristics of connective tissue. This slide confuses students, so let's pay very close attention to this and how I say this. Connective tissue tends to be well vascularized. Connective tissue is well vascularized. Lots of blood vessels in connective tissue. You cut it, you injure it, it bleeds. If you cut bone surgically as an orthopedic surgeon, it bleeds like crazy. But it's also why it heals so well. Okay, because blood is necessary for healing. And so if you have a well vascularized or a well perfused with blood tissue, it's going to heal very quickly. The exceptions to this rule, there always are, right? Remember this, like in biology and physiology, things are typically not never or always. There's always seems like there's always like a, exception. So the exceptions to connective tissue being well vascularized are cartilage, tendons, and ligaments. And that's why, how many of you are athletes and have had an injury to one of these three structures? Let's see it. How well did it heal all by itself? Yeah, it never healed, right? Or I had to have surgery, or right? They had to replace my ACL, my anterior cruciate ligament. I had to have it reconstructed or replaced. With a bone, how many of you have broken a bone? Okay. How well did it heal in like 8 weeks? Yeah, it was like fine. That's the big difference because of the vascular supply. Okay. So the exception, cartilage, tendons, ligaments, not well vascularized. Another component of connective tissue in the matrix, the extracellular matrix, that makes it really, really important for us to pay attention to is this gel component. This gel component, because remember we used the Arrakis example of how, the dune Arrakis example, that water is so precious because we're like 50 to 75% water, right, as an organism. And a lot of that water is in the extracellular matrix. It's in the connective tissue outside of the cell. And water likes to move. We've seen that, right? Water likes to move to where the solutes are. Well, we already talked about how the extracellular matrix has a high concentration of sodium. because of that sodium potassium pump pumping a lot of ions out. But the inside of a cell actually has a lot of solutes, and we want to make sure that it doesn't, swell and rupture. So how do we keep the water in our tissues? Well, this funny looking structure here looks like a millipede in the upper right, and then if you zoom in on kind of the backbone of this funny word known as a proteoglycan, you zoom in on this box and you look at this structure that looks like it has like bottle brushes on it. This is the secret. So this gel component is known as a proteoglycan. This is a protein sugar. This is a polysaccharide. You know what that word means now. This is a many sugar that's attached to a protein. And that makes this structure known as a proteoglycan. And this proteoglycan carries with it a net negative charge. Net negative charge. So we have a lot of sodium that we pump out of the cell. You remember that? The sodium potassium ATPase. A lot of sodium goes out. 3 go out and 2 potassiums come in. 3 sodiums go out, 2 potassiums come in. Every time you run it, you spend an ATP molecule pumping out 3 sodiums. Well, this net negative charge in this proteoglycan, which is the gel component that's in our matrix, is slightly negatively charged, and so it attracts what? Sodium, which is positively charged, Na+, forms a bond. What does water like to do to an area of high concentration of sodium? Water follows, right, through osmosis. And so the tissue in our body operates like a sponge. You have a polysaccharide binding to a protein, makes a proteoglycan, has a net negative charge. Sodium is being pumped out of cells. It is attracted to that net negative charge. It binds. Water follows. And now you have a sponge-like structure that has a lot of hydration in the tissue because of this whole cause and effect of all these molecules, this domino effect. That it took me a couple of weeks before I could explain this slide to you, and for you to actually have, recently heard all these things to understand mechanistically how that works. Does that make sense? You have a many sugar, a polysaccharide, attached to a protein. That's what makes up that proteoglycan. Look at the scale here. This is 1 micron. 1 micron. So this is maybe a 3 micron structure. And here's the core protein. Here's these glycosaminoglycans. These are the sugars. And this attracts the sodium because it's a negative charge. Water follows sodium via osmosis. Now you have a tissue that's well hydrated that acts like a wet sponge. So it resists compression and it creates space and openness in your connective tissue. Kind of cool. So these matrix components outside, other than the gel, we've got a fibrous component made up of 2 structures. We have a fibrous component made up of collagen. This is the most abundant structural protein in the body. About 25% of the total protein that's found in all of the body. And there's 20 plus different collagen genes. There's many different varieties of collagen. And together, all of these collagen bundles make these fibrils make these bundles. And we talked about this. It's like a braided thread. And so you have a collagen fiber that is made up of braided strings of collagen. And in this areolar connective tissue on the far right, that's the larger diameter structures that are slightly pink in color. But if I was to black and white this image, now that we've talked about the shapes and the microarchitecture, I would bet you could still tell me which were the collagen fibers, right? And the darker squiggly lines are what? Elastin. Elastin is a long thread-like fiber makes up the minority of the space. Collagen's the majority of the space. But the elastic fiber is like an elastic rubber band. And so when the tissues have stress on them and they actually are stretched, the elastic fibers give it the recoil that bring it back to its starting position. So this is why all of you do this. You grab your skin, you pull on it, and then you let it go. What happened? You're like, oh, wow, it went right back to where it was. Okay? That's the elastic fiber in your skin. Now, as you get older, your elastin replenishment diminishes in your tissues. Collagen continues. The gene for collagen continues. The gene for elastin actually turns off as we age. And that's why our skin, as we get older, doesn't look the way your skin looks right now, unfortunately. Okay? This is the best it's going to be right here, folks. Okay? As you get older, that little experiment doesn't work so well. It works less and less and less. Okay? So elastin, what gives us this elastic recoil? And just for sake of completeness, so the red arrow is pointing to a collagen fiber. The green arrow is pointing to an elastic fiber. And then this smaller, thin, thread-like fiber that's about half the diameter of an elastin fiber is a reticular fiber, the yellow arrow. So we have different combinations of gel and matrix, right? We could actually have elastin greater than collagen in a tissue, and you have more elasticity. Like you're going to see this in the bladder. You have a lot of elastin in the bladder. Thank goodness, right? Collagen greater than elastin, more tensile strength. Tensile strength means a resistance to fracturing. It's stiff, but it's strong. If you have more gel over fibrous, right? Because collagen and elastin are fibrous protein examples, structural fibrous proteins. If gel, the proteoglycans dominate, then you have more water content and you have more cushioning, like you see with cartilage. More shock absorbing characteristics. So do you see how like the different makeups of the tissue give you different functionalities and purposes that make a lot of sense when you think about, oh yeah, that's why my cartilage is so spongy, because it's like a shock absorber. Okay, are you guys comfortable? I want you to be able to compare and contrast the characteristics of epithelial and connective tissue for exam 1. So do you want to try a couple of examples of histology? You may see some of these examples on the exam. These could be potential test questions that you might see a week from today. It might look exactly like it's looking right now. Okay? Just saying. You have an answer?
Oh, no.
Oh, question.
Will the test, like, thing be in color, or will it be?
The test packet's in black and white, but the images will be broadcasted on the screen. Let me repeat this. Let's practice this because some of these pictures you might see exactly like you see them now. Does that make sense? Yeah, I don't know how I could be like more obvious. I'm really trying to be helpful here, folks. So let's study these together. What do you think this is? Stratified squamous, E. Well done. Well done. You can see the top layer, right? These are squamous epithelium. They're wider down here at the basement membrane. Okay, how about this one? I heard it over there. Who said it? It's dense, irregular connective tissue. So it's dense. You hardly see any nuclei, right? So you know it's connective tissue. And it's densely packed and irregular because all of these lines are going this direction. These ones are going this way. This one's going down here. This one's going this way. That's irregular. Okay. How about this one? And you're going to have more of this in lab this week. More of this like reinforcement of being identify different types of tissue. It is the hyaline cartilage. It's the hyaline cartilage. That's right. Hyaline cartilage. Okay, any questions before we move to the last